So, afternoon, everybody. Thank you very much for coming along. This is the last, I mean, it might be the last round. I don't know if there's a grand round next week or Christmas. Anyway, it's the last DEM for 2024. DEMS is the Devon and Exeter Medical Society. I'm Tim Malone. I'm currently president this year. And we have meetings in the evening, and for the last year or two, we've had meetings at lunchtime every couple of months, taking over the grand round slot. So to bring DEMS to a new audience, I encourage you, when you become more senior in new medical careers and stay in the area, to join DEMS. So if you're a consultant or a GP in the audience, please consider signing up for DEMS, because that's the funding behind DEMS. It's a charity, medical charity. But the meetings are free and open to everybody. And I always have to say this, and everyone will smile. DEMS, Devon and Exeter Medical Society, was founded in 1780. So it's been going a while. This is the second oldest medical society in the UK. The London Medical Society was founded in 1773. London Medical Society have a grand building, which we saw recently, went up London and found it. Doesn't look as though there's anyone in it at all, actually. They're completely deserted, but they have a grand building. Yeah, we don't have a building at all. But anyway, we are the second oldest medical society. So great to have you all here. We're recording the meeting. You can hear us online, I hope. And my pleasure today is to welcome my colleague, who I worked with for the last few years on global and planetary health, teaching at the medical school, Mick Voss. And Mick is a marine microbiologist, or microbiologist by training, but has a great interest in oceans and the marine world, and is a very keen snorkeler and a very keen underwater photographer. And when we were, my wife and I were down in Falmouth earlier this year, took us at rock pooling, and I just I had seen things in rock pools that I would never ever have seen before. So, expert guide on rock pooling, and his son actually is an expert guide as well. We were seeing, God, I never noticed. So good. Mick did his training in Rotterdam, then his PhD in Germany, and moved to Oxford for a while, and then came down to University of Exeter.
Yeah, I was in Holland in between as well.
In between, so yeah. But welcome anyway. It doesn't often come up all the way and out of the country, different country of Cornwall into the Shires. But welcome to the Shires, Mick.
Yeah.
Oceans and human health.
Thanks for the introduction. Always good to be in the oriental lands of Devon, I say. Thanks, Tim, for the lovely introduction, and also for taking me out to a really nice dinner. Yes. So yeah, my name is Mick. I'm based on the Cornwall campus in Penryn, and Tim asked me to give a presentation, which I had to scratch my head around about a little bit. So Tim and I, as he said, we're teaching the special study units for the last 5 years, which is online. So we're both passionate about planetary health. As you know, Tim is just very passionate about planetary health. So I wanted to mention that in my talk. Tim asked me, as he also already said, to show me some pictures of marine life in Cornwall, which I will do, but I have them at the end. So if you have a phobia of crabs or fish, then you can leave like at 2/3. Or if you have a phobia of microbiology, you can switch off and then when I show those. And also, I'm a microbiologist by training. I did my PhD actually on this Mixococcus, not named after me, but it forms these multicellular fruiting bodies, so you can just about see them with the naked eye, and they're actually produce antibiotics. So I had to make a talk out of these kind of disparate fields, And I think actually I found an angle. So there is actually overlap. So planetary health and marine biology, there's an overlap in kind of the burgeoning field of oceans and human health, which was started by or co-started by my mentor and past leader of the European Centre for Environmental Health, Centre Director Laura Fleming, who's now retired and potentially online. I can't see that. So that is kind of the connection between planetary health and marine biology. There's also a clear connection between planetary health and climate change and infectious disease. This is an example of Vibrio bacteria, not the cholera bacteria, which also has a strong link to climate change, with vibriosis, non-cholera bacteria that cause gastrointestinal illnesses and skin infections and have a very high mortality rate, which thrive in warmer waters. So that's clearly linked to climate change. And then there's also a link between marine biology and microbiology through some work I did with students on seaweeds and the antimicrobial compounds that seaweeds produce. So that's what I'm going to talk about today. So first, kind of the ocean. I like this map, Spillhouse projection, which kind of highlights the fact that the ocean covers 71%. Bit of a cliche to say that it's strange that the planet's called Earth. Well, it's clearly water and so on, but I think this is a nice illustration. And I also like this illustration to show that a significant proportion of humankind lives very close to the ocean. So it's a Pearl River Delta mega city with some cities you will have heard of, such as Hong Kong and Shenzhen. And there's also a whole bunch of really, really large cities that you probably have not heard of. I guess I'm from a mega city a little bit as well myself, because Rotterdam where I'm from, and Amsterdam and Utrecht and The Hague are all connected into an agglomeration of 7 million people. And not only is it close by the city, but it's below sea level. So there's a connection there. I put this slide here. I thought maybe Tim would appreciate this. So I'm based in Cornwall, where tin mining was a major thing up till a couple of decades ago, and then there's still plans maybe to start mining certain minerals. And people from, all the way from the Middle East, the Phoenicians visited Cornwall many thousands years ago. And according to this publication, Jesus was one of them, together with his uncle, Joseph of Arimathea. So I think it's true. What do you think, Tim?
Yeah, it's going the wrong way. It looks like a corner.
Yeah, the tin went that way, and it probably went around this way. But yeah, it is in the science section, so I think there's probably an element of truth in there. So there's also
the mail online. So
yeah. So the ocean has facilitated kind of exchange of goods and ideas. Of course, also less nice things. Sunday, I was in the Pitt Rivers talking about colonialism, which I can still recommend visiting.
Cool. It's tuna.
Sorry, we have lost the speaker's sound. Hi, Tim. We cannot hear the speaker.
Hi, sorry, can't hear the speaker. Sound has gone.
Can you hear us now?
Can you,
Yes, we can.
Okay.
can you hear me if we talk, we use this?
No,
No, you don't need to
it's
do that
muted.
here. Okay. So actually you have to use this one. All right, I think, I don't know what I did. I don't know if it's possible to accidentally mute yourself. Can you hear me?
We can hear you.
Okay, great. I'll continue. So this slide, yeah. So this is something that is very current now. It's all sorted, Mark. We need all kinds of metals, manganese. It's sorted. It was muted. Which we'll get from the sea floor probably in the next decade. But there's a negative side to that as well, because we're destroying kind of organisms we probably even haven't discovered yet. Yeah, this is a nice picture of a giant floating norovirus vessel. So the ocean, of course, is also very popular for leisure. Again, there's negative consequences as well. Enormous carbon footprint and sulfur pollution, noise pollution, light pollution, strikes of whales and all kinds of things. Introduction of invasive species. When I teach ocean and human health, there's always students giving presentation on the benefits of wild water swimming, both physical benefits and mental health benefits. And I absolutely believe that they exist. But as you know, there's also a risk of drowning, actually a major cause of mortality, about 250,000 people per year globally. And I don't have to tell you about Southwest Water and how they are managing our waterways and our sea. This is actually a colleague of mine, Dr. Anne Leonard, who did a beach bum survey where she swabbed, not personally, but she asked people to provide rectal swabs, surfers, and paired friends that were not surfers, and the surfers had more antibiotic-resistant bacteria colonizing their gut, presumably because there is a lot of water ingestion when you're surfing. So that's the danger. And this is a summary of all these kind of positive and negative effects and all these various pathways in which the ocean can influence our health and well-being by Laura Fleming and colleagues. And you see, it is tremendously complex, and it is a mix of kind of negative impacts, and you have this kind of classical exposure and negative health outcomes paradigm, but there's also this kind of newer, more positive paradigms that you can actually benefit in terms of your well-being and health from your contact with the natural environment, including blue environments. And this is kind of a simpler version for my students of that same graph. So there can be, you can get vitamin D, or you can get sunburned, you can get physical exercise, or maybe eaten by sharks. So there's positives and negatives from our interaction with the sea. So a little bit about my own research about health in a very broad sense, and the sea. So when I got back, or when I moved to Cornwall 12 years ago, I kind of rediscovered my love for marine biology, but more as a hobby. So when snorkeling often, and it's really amazing how beautiful it can be on a nice day in spring, looking at the seaweed. So I thought I need to find an excuse to go snorkeling in the boss's time, which I did. So first with the following student, Abby Kolklove, who was a medical sciences student, and she had a 1 year kind of a placement training year with me. And we went, so this is at the beach at Falmouth. We went to Falmouth to collect seaweeds and have a look at seaweeds. And the reason is, or motivation, was the rise in antibiotic resistance, which I probably don't have to tell you about. I quite like this graph with on the top the year of antibiotic deployment of several main classes of antibiotic, and then here at the bottom the years that the resistance to that antibiotic was first observed. So you see that bacteria quickly evolve resistance to antibiotics because they have very large population sizes, very short generation times, and they also have the ability to swap genes between them, including resistance genes. So that's a problem, and it's especially a problem because we're running out of good antibiotics. So there's like a gap in delivery, and there's probably multiple reasons for it. I guess the main reason is that it's not very profitable to develop. It's hard work, costs a lot of money. It's probably not prescribed for very long if it is finally prescribed. So there's not a lot of money in it. So seaweeds are known to be very metabolically diverse and have lots of secondary metabolites with antibacterial and antiviral, anti-protozoan activity. So that's been known for a long time. So the good thing is that in Cornwall, or in the southwest in general of the UK, we have an enormous diversity of seaweed. So this is just a patch of a couple of like half a meter, and you can see lots of different species. There's lots of smaller species that I haven't bothered to ID, but you consider there's an amazing diversity of seaweeds, including metabolic diversity. This is a slightly larger scale, so this is a couple of meters with kelp, and again you can see you have all these different species growing side by side, especially in spring. So this is what we did. So I had my excuse to go scuba diving in work time, and I collected seaweeds, brought them back, rinsed them, put them in the freeze dryer. Then I got the James Martin coffee grinder to grind up the dried seaweeds. So it was a tool. We had to buy a second one because it broke because we processed too much. So you got these different dried seaweed extracts that you can dissolve in methanol, and then you can dunk these little diffusion discs in them, and then put those in the Cat 2 hood to let the methanol, which itself is toxic, evaporate. So you have these discs impregnated with a portion of the secondary metabolites in seaweed. By the way, if anything is not clear, please let me know and interrupt me. So we had seaweed extracts, and we paired those with bacteria. So I went to colleagues in the Royal Cornwall Hospital, and they supplied us with Staph aureus strains from actual human infections, all different, or with different antibiotic resistance spectrum. I don't have to really introduce MSA, I think, to this audience. And so the experiment we did is you can grow the different strains, and you mix them with agar, and you pour them in a petri dish, and then you can incubate them overnight, and the bacteria will grow in the agar, turning the agar opaque. But what we also did is placing with forceps these different discs with different seaweed metabolites on the agar, and then overnight the bacteria grew up, including around some discs, but not other discs because they had antibiotic properties, so they couldn't grow. So this is a way of quantifying resistance or susceptibility to antimicrobials. So this was the experiment. We paired all the seaweed extracts to all the different Staph aureus. And indeed, we found that there was a lot of antibiotic activity. More than half of the extracts showed activity on at least one of the Staph aureus strains, and that included the three main seaweed types: green seaweeds, you get red seaweeds, which are kind of plant-like, and kelps, which are brown seaweeds, which are more related to potato blight, actually very different evolutionary, but they're lumped in with the seaweeds. So all these different major seaweed types had staph activity.
Mick, could I just ask there, how do you know that the effect isn't because the seaweeds have been exposed to antibiotics from Southwest water?
Yeah, I guess we had controls.
Okay.
Well, actually, we don't have, no. So we had controls where, of course, the discs were with methanol controls, but not clean and potentially exposed seaweed controls. So I mean, I don't think that's likely, but you're right. I can't exclude. I don't have the data to exclude it. So usually when people try to find antimicrobials, they take lots of organism, plants or seaweeds or other things, and they call that kind of grind and find. They grind it up and they expose it to a panel and see what works. And we want to do it a bit more smart, so ask some kind of ecological questions. A lot of seaweeds are invasive species, so we tested whether invasive species maybe have properties that make them more kind of toxic, which was not the case. So that was kind of something to know. You don't have to really go out looking for invasive species to find most activity. These are three species that are closely related. So we thought, well, if species are closely related and maybe their antibiotic profile, antimicrobial profile is also closely related, then it makes sense to sample kind of more widely and not focus on related species, which might be the same. And that was not the case. So there were two species. So you got here a tree of all the Staph aureus, and you got three seaweeds here, and two are indeed very similar in their activity patterns, but another one was very different. So that kind of, is also something that people can kind of keep in mind. You can sample closely related species and potentially find different antibacterial activities. And this is actually in the middle one. That's my favorite seaweed. So this is a photo I took of bushy rainbow rack. So you can see it changed its name a couple of times in the last 10 years. And this is a species that is really bright blue. You can't miss it if you go here to the shore in rock pools and you see something purple or blue. First, I thought it was kind of oil slick or something, but it's the actual color of the seaweed. And it's not a pigment. This is caused by a process called iridescence. So it's the same as on butterfly wings or oil slicks or CDs for the older people in the audience. So it depends on the angle of view what color comes off it. So that's really striking, and you don't really have this effect if it's out of the water. When you submerge it into the water, it turns this really amazing blue or purple. And just as an aside, I thought maybe Tim likes a bit of background. So this is how this was described by an eminent naturalist who lived in Devon, actually, and the inventor of the word aquarium, Philip Henry Goss. And this is how he described this effect of it being not so nice above water, but really beautiful underneath the water. So that was, I think, very nice way. That's not really how we write about nature anymore. Instead, in 2018, before I brought Heather Whitney, who was at the University of Bristol, who works on iridescence in plants, and I told her, do you know about this seaweed? And she didn't. So I said, come down to Falmouth and I'll show you this seaweed, and you can try to figure out what it is. And she was very enthusiastic because, of course, it is very striking. And she published a very high-profile paper where she found out exactly how this mechanism works, which was really nice. That's the bushy rainbow rag. And finally here, what we did, we had these data, Vitec data on clinical resistance. So we could kind of score the amount of different antibiotics each Staph aureus was resistant to, and also how many of the seaweed it was resistant to. And in general, you see that there's a positive correlation. So things that are multi-drug resistant are also better at withstanding all these seaweed metabolites, which, by the way, we don't know what's in these extracts. It's a mixture of lots of things. I'm not a chemist. There's probably thousands of molecules. So exactly what the active compound is or compounds, that's kind of a black box for us. And this pattern, this kind of positive correlation of resistance to two different things, is called cross resistance, which you might have heard of. So if you have a cell, and for instance, it has a membrane pump, and it can pump out antibiotics, that same pump can be used to pump out metals, for instance. So you are cross resistant, resistant to both these things. And that could be because of co-selection. So it could be potentially that if bacteria settle on seaweeds and evolve resistance to the seaweed, it might also give them resistance to clinical antibiotics, or the other way around. So if you have, like you say, resistant strains from the hospital entering the water, those might be the bacteria that stay alive when they settle in the rock pools, because they can survive the onslaught of the seaweed antimicrobials. We couldn't really answer that question using in this study, but we did another study where we could kind of address this phenomenon of co-selection directly. And this was not in the ocean, but it was in the pond, so it was in the water, not in Cornwall, but on Merseyside, which is a region with a lot of kind of historical pollution events, as you can see. And instead of what most studies do is they compare a polluted site with an unpolluted site. But we took a different approach. Instead of a spatial approach, a temporal approach, where we took one site and tracked pollution through time. And we did that because we could do that because we acquired a sediment core, which is kind of an archive through time. We didn't collect it ourselves, but we played around in Cornwall with a macro core that you can see here. So you can drop this down, and with compressed air, you can punch it into the sediment and also make it float again, which is quite dangerous. So Macroth, who invented it and played around with on Lake Windermere, was actually, his boat capsized when the core came up, and he fell in and he got pneumonia and he died. So that was also an interesting link to microbiology there. Anyway, so that was fun, but we got this core, and my student Andy Dickinson and my rest student sliced the core in 51 centimeter slices, and he did all kinds of measurements. He together with colleagues, so he did some sequencing, and he measured different metal concentrations throughout the core, and we also managed to date the core. And this is the age of the core, which is actually almost 200 years old, which surprised me. So it's a really kind of nice, long historical record if you go down into this sediment archive. So what we did is we quantified zinc as one of the main metals, and you can see that there's a difference in concentration through time. What he also was able to do is revive bacteria from the core. So culture very, 100-year-old bacteria. They were there just kind of in very cold, maybe anoxic sediment preserved. So you can plate them out on agar without anything, so everything grows. These are all colonies that he picked and then subsequently plated out from a 96 well plate. You can also make agar with zinc. So things that are zinc resistant will also form a colony, but things that are not resistant to zinc will not grow up on the plate because they can't. So you can kind of score how many out of 96 colonies are zinc resistant. So you have like a zinc resistance profile of the different bacteria, but you also plate them out on cefotaxime, which is an antibiotic. And you can see that there's a really nice kind of correlation between resistance to zinc and cefotaxime. And the interesting thing is that cefotaxime was only introduced in the clinic in the 80s, but we see that bacteria are already resistant before it's introduced because they have these metal resistance mutations. So I thought that was a nice illustration where we could more closely connect how human anthropogenic pollution can select for antimicrobial resistance. And of course, so AMR is very important. We're very concerned about bacteria developing resistance, but we should be most concerned by bacteria that are actually pathogenic and virulent. Those bacteria being resistant, because those are the bacteria that we would like to treat. And so coming back to tin in Cornwall, as in my earlier slide, we're here in Cornwall, so this is not too far from campus. It's one of the most polluted sites in Europe. It's full of metals. So if you go walking here, there's a BMX trail where the soil is about 3% arsenic. So it's really polluted. There's nothing swimming in the Carmen River. And that was mainly the result of a tin mine that was closed in the 90s because the tin price dropped. And then the mine, which is more than 400 meters deep, fills with water. The water gets laden with all these metals, and then a dam broke, and in the 90s, all this water, contaminated mine waste, flowed into this river and into the sea in sediment. So that's kind of the reason why it's so polluted. So we had a look there, and we asked the question: can metal pollution, it can select for antibiotic resistance? Can it also select for virulence, which had not been studied before? And so why would that be? We had an idea, and I hope I can make this clear using this schematic. So these are some green Pseudomonas cells. And what Pseudomonas in an infection is always very limited by iron. And
This is mouse to point to.
okay, well, yeah. And so our body actively kind of withholds iron, so infectious bacteria have trouble growing with things like transferrin and other molecules. So these bacteria have evolved mechanisms to scavenge for iron, and these are called siderophores. So they secrete siderophores. Siderophores have very high affinity for iron, and then they're able to take up the siderophore and the iron using receptors. So they scavenge iron, and then they can grow better. What these siderophores also do is they bind to non-iron metals, including these toxic metals like zinc and copper we looked at. However, they cannot be taken up by the cell. Of course, they don't want to take them up because they're toxic. So our idea was that if you expose bacteria to toxic metals, they start to evolve to produce a lot of siderophores to detoxify their environment. But because they produce so many siderophores, they're also better able to sequester iron, and they're more virulent. So that was the general idea that Luc, my PhD student, sought out to test. And he used experimental evolution, which a lot of people in RIN do. So bacteria, like I said, a very short population, large population sizes, short generation times, and you can keep them for hundreds and hundreds of generations. You can passage them every day, and they grow, and there's many generations. And so you can track evolution in real time. You can replicate it, and you can force them to evolve in different conditions. So he took a clone of Pseudomonas aeruginosa and evolved it without copper, or a low concentration of copper, and a high concentration of copper for 12 days. And then what he did, he measured whether indeed siderophore production could change in that time. There was selection for more siderophores, and also whether that increased virulence. And if anything's not clear, please give me a shout, because this might not be something you've heard of before. So these are his results for pyoverdin, which is the main siderophore. And let's focus on the right panel, which are the results after 12 days of evolution. You see the 3 different treatments at the bottom: no copper, a little bit of copper, and a lot of copper. And you can see that there's a clear, significant trend of producing more pyoverdin at higher copper concentrations. So that kind of fitted our expectations. Then he wanted to know whether, because of these higher therefore production, these bacteria were also more virulent. And for that, I don't know if you've heard of it, we used wax moth larvae, Galeria melanella. So a lot of studies use mice, but they're of course it's very expensive to run a mouse facility. I don't think we have one in Exeter. There's all kinds of ethical issues with killing mice as well. And this is actually a really good model system. So you can take these caterpillars, you can just order them online because people do that to feed their geckos and so on, very cheap. You can inject them with bacteria, and when these bacteria are virulent, they turn to mush, literally. And you can quantify that, so you can take 20, inoculate them, and then kind of look at the survival rate. You can incubate them at 37 degrees, so that's a bit like us. And the innate immune system of insects, they don't have an adaptive immune system, but the innate immune system is similar to ours. So things that are virulent in mice are also virulent in these guys. So we chose to do that. So he injected these different evolved treatments into groups of Galeria to produce these kind of death curves or survival curves to see which bacteria were most virulent. And again, let's focus on the right panel after 12 days. You can see that if there was no copper sulfate, and they also didn't produce as many siderophores, the bacteria were less virulent than at higher copper concentrations. So he showed that actually metals can select for virulent bacteria, which is a bit of a worry, if there's lots of metal pollution, such as in common. Virulent and resistant bacteria. Moving on to another student project. We know that Pseudomonas is widespread in the environment, but we're often wondering, okay, so what other things are in our soil and in our water, marine water, fresh water? The gold standard to look for microbial safety is to count the number of E. coli. So you take some water sample, you put it through a filter, you put a special expensive agar where E. coli cells turn blue, and you count them, and you have a concentration. The concentration, if there's lots of E. coli in water, it's probably not water that you want to drink or swim in. So that's basically it. But there's of course also a lot of other bacteria, and you wouldn't pick them up. Some other gut bacteria, of course, will be expected to be correlated with E. coli, but things like Vibrio or Pseudomonas have very different lifestyles, so there's no way, and you don't want to use a different agar for every different pathogen. That would be a little bit too much work. This is Campylobacter agar, and this is Salmonella agar, also very expensive. So we had a brainwave, and we thought we use these wax moth larvae, instead of injecting them with clones, we take a whole water sample and inject that into the Galeria. And if there's nothing dangerous in the water, then they'll be happy and live. And if there's some dangerous pathogen in, then they will die. And then we can try to figure out what the pathogen was that killed them. So that's what we set out to do. So the student, Rafael Hernandez, visiting a summer from Miami, and we took 2 trips through 8 sites, beach and harbor kind of sites around campus. And we took the sediment or sand, we washed it. So we had kind of the sediment wash, we concentrated the water, and each sample was injected in 20 Galeria. And then we produced those survival curves to see which samples produce mortality. And importantly, we also used controls. So we filtered the water with a filter small enough to exclude bacteria. So if there was also mortality in the filtered controls, then that was not due to bacteria, but maybe through pesticides or other things. So these are the results for the 2 different dates, and for the sediments and for the water. And you can see that there's very different trends for the different samples. So with a lot of samples, you see that there's not really an effect. So you see here, there's straight lines, so everything survives. But there are also a couple of samples where you see that over the course of 3 days, lots of Galeria died. There's only 1 out of 20 left. Importantly, with the mortality controls, nothing died. So we know this is caused by something bigger than the filter size, so presumably a bacterium. So we wanted to know what was killing these poor Galeria. what was in the beach sand that was so dangerous. So we selected the 4 samples with the highest mortality, reinfected the Galeria again, then we plated out the innards, kind of the lymphatic juices of the larvae with all the bacteria growing in it. And then we picked a single clone, which would be the thing that was killing them, and injected those clones, like Luke did, clone by clone in Galeria, either 110,000 or 1 million. And then we sequenced those clones to find out what they were. And we found some, to paraphrase Donald Rumsfeld, some things that we expected to find, and also some things that we did not expect to find. So first, you see something that's very virulent, Proteus mirabilis. You see everything's dead after 12 hours. So that was pretty nasty. And we sequenced the genome. We found that it had a salmonella genomic island that it acquired through horizontal gene transfer. It was found in China and in France, in hospitals and farms, but not to my knowledge in the UK. Then we had something that was less virulent, and that turned out to be Vibrio ingenesis, which I'd never heard of. And I looked in the literature, and it was only reported once before in 2 septicemia cases in South Korea. So it was discovered in a very severe infection in South Korea, and that was it. But we also found it on Gilly Beach in Falmouth in the sand. So that was interesting to see. That's not something that we would have otherwise noticed. Then I guess the usual suspect, E. coli, also quite virulent, with more than 200 virulence genes, including virulence genes that are known for very nasty different types of human infections. And then finally, Pseudomonas aeruginosa again. And this one also had lots of virulence genes, but also almost 50 different antibiotic resistance genes, also some that looked quite significant. So there's clearly a lot of dangerous bacteria around us in the water and in the sediment. So should we be worried? I guess most of us are not regularly infected by bacteria, hopefully. And you can see this is very, very outdated. But through various public health measures and the invention of antibiotics and vaccines, infectious disease tends to not be such a big problem anymore. Although, of course, this could change with the antibiotic resistant crisis, which would make standard procedures a lot more dangerous, and also maybe climate change. A lot of examples of climate change and pathogens, a bit of kind of sensationalist almost sounding ones where the permafrost is melting and dead animals come to the surface again and release their anthrax spores. You get extreme weather events, and you get aerosols, and you get Legionella cases being linked to extreme weather events, as well as these Vibrio bacteria that I mentioned before. And kind of that inspired us to, clearly there's lots of pathogens. There might be an increase in incidence of certain pathogens, but in order to study that, we need to know what the diversity of human pathogens is. And there wasn't really a resource for human pathogens. I mean,
Fine.
yeah. So another poor medical sciences student did literature research for us and found that there were 1,513 species known to infect humans, which is quite a lot, quite a wide diversity, and also an increase in the description of new species. And we actually described, again, Mail Online, authoritative medical journal. We were covered when we described Staphylococcus cornuliensis, or our own Cornish pathogen with colleagues at the Royal Common Hospital.
Is that the only journal you can get it, Henry?
Yeah, it's certainly the most read journal, I think. And so this is the very final science thing I want to show. It is obviously a lot of work. You can't have students scouring through the literature. We saw that mistakes were made, both false positives and false negatives. So we got talking to Fabrizio Costa, a colleague at the Institute for Data Science and Artificial Intelligence at Stratham, and say, let's have AI do it for us rather than a student. And that's what we have been doing. So we take all the known bacterial names and look for them in all the abstracts indexed in PubMed. we filter that. So we have the abstracts that mention patients or humans or children, elderly, or sepsis or infection, anything that has to do with infection on humans. And then we have all these abstracts, tens of thousands of abstracts, which you can't read or interpret. That's too much work. And we use a large language model. So you must have heard of ChatGPT, and say, here are the abstracts. Tell us if the people writing the abstract regard the bacterium that is mentioned as being the cause of disease, which is difficult because there's lots of bacteria that are from clinical samples. But if you get a bacterium from a sputum sample, it might not be the causative agent of disease because it's not a sterile site. So there's lots of different bacteria in there. And this is kind of what it looks like. So you give a prompt. You have to be very careful about this, and you have to say, you must say yes or no only, because these things can also hallucinate. They can go a little bit off piste sometimes. So you have to be careful. So there was a lot of thought involved and a lot of checks involved. So we instruct the AI. Also, the bacterial taxonomy. If you were thinking of going into bacterial taxonomy and nomenclature, can I advise you not to do it, because it's not a lot of fun. A lot of bacteria have like 6 or 8 or 10 different names. So we needed to make sure that we merge results for all those different names into one book. And so what we produced is a pathogen dashboard where you can click on all the hundreds of pathogens and find the PubMed evidence and also the interpretation of the LLM about the causative nature of the case. So for instance, if you go for this species that I mentioned, it has 55 results, and you can click and go to the PubMed article, and it gives you the title and gives you why it thinks it is a pathogen. So that's something we're working on and will provide as a resource for the global microbiology community. That's all I had, and I don't know how much time we have. I guess if you're not really interested in things like that, then feel free to go. You can ask me some questions about the science, and for those who are interested, I can show some of the marine biology photos. Thank you.
Show us some photos. Yeah, that's fantastic. Yeah.
Or any questions? Yes.
So
Do you think we should now start changing our prescription habits from pharmaceutical antibiotics to more seaweed prescriptions?
I don't know. Like I said, this was something we didn't follow through with going into the chemistry and then the pharmaceutical side of things. Actually explored it, but then I quickly retreated because that was too scary. I'll leave that to other people. But who knows? Yeah, there are interesting compounds that we don't know of, and could be the next antibiotic. Yeah.
You already do that one, first of all, didn't you?
We do.
Yeah, yeah.
We just need big bags.
Yeah, okay.
I just had a question about sort of how those antimicrobial properties would be there. Like if you were to ingest the seaweed, like if they're edible, would you then have them?
There's definitely, you will see effects on your gut microbiome with these different diet interventions, and then you have probiotics, but you have also prebiotics, and I think seaweeds are an example of that. I mean, seaweeds are very rich in fiber, so definitely very good for gut health. And I mean, it's not a lot of research, but we know that people in Japan, where they eat a lot of seaweed, generally tend to be quite healthy and long-lived. So, yeah.
What's the pictures?
I don't have the question online.
Any questions online?
Could I ask about mussels and oysters sold in shops? Are they screened for bacteria before they arrive in supermarkets?
Yeah, they're depurated, so they keep them in clean water so they can expel dirty bacteria. And they're also monitored also for harmful algal blooms that can close kind of shellfish companies for a while, because then that's a toxin. But yeah, I think with increasing water temperatures, there might be more problems with Vibrio bacteria, and they're very nasty bacteria, unfortunately.
Could I also quickly ask you, do you know Ian Hendy?
No.
No, I was flying to Vancouver a year ago, and I was sitting in a cafe in Heathrow Airport, and I saw this guy who sat next to me with massive, great biceps. And I started talking to him and asked him how he got his biceps. He said he'd been a world champion bodybuilder, but he was also a marine biologist. And he works in Southampton, and he travels all over the world dealing with polluted marine environments and what to do about it all.
I'll look him up.
Yeah, he's a really interesting guy.
Who's asking the question?
I'm sorry, I'm Hugh Sheridan. I'm a retired GP, but since done a bit of forensic training since I retired.
Thank you very much. Thank you.
Okay.
We'll show some pictures now, and if you've got any more questions.
I'm conscious that we only have 6 minutes of the hour left, so feel free to just leave.
Let's show. Yeah.
So anyway, so I started a website. I was talking to a Cornish PhD student 10 years ago when I started putting my photos online. I want to have a Cornish name. I said, what is a really Cornish kind of rock pooler name? And he said rock pool hunter. So that's a unique characteristic of the Cornish language, that is Essor is hunter of, and then it's hunter of rock pools. So I thought that was good. I think it's a nice name, although no one ever remembers it. So I said, oh yes, and on Bolognese, I said, oh nice. And then 2 seconds later, of course, they don't know. They don't know what that name was. Anyway, you can see some of my pictures there and more on Instagram. Rock pools. I guess this is not the best time of year for rock pooling, but the only nice thing I think of winter is that there are storms, so you can go beach combing, which is really nice thing to do. Just kind of blow the cobwebs out. And this is something we found 2 weeks ago, which is a grey triggerfish, which actually swims around in summer, but they're not very good with the cold, and then they wash up sometimes. But divers see them off Plymouth relatively regularly. It's more of a southern species. And if you
put it in your hand, if you've seen one of those, anybody? One. All right, fantastic.
Yeah.
Very common on the Yelm River, and kids were catching them. And I'm very surprised because I've seen these in tropical waters, and I just wondered if it's a global warming issue.
Yeah, well, that's kind of that would make sense. Warmer temperatures. Yeah. If you're interested in beachcombing, I can, by the way, really recommend that book. It's not only biological finds, but also human debris that you can find, which is interesting. So yeah, one other thing last year that my son found. So we were at Sennett Cove in the west, and there was really nothing on the beach. So we're a bit disappointed driving all the way. But then he spotted this, and he knew exactly what it was from a David Attenborough documentary. You might not know, but it is an amphipod still crawling around, a little crustacean in a salp, which is kind of a swimming sea squirt. So what it does, it finds a salp and hollows it out while it's still alive to lay its eggs and rear its young, floating through the deep sea. So I gave this to my friend David Fenwick, who is a kind of amateur naturalist, and I can recommend his website if you want to look up your finds. And he took these kind of lab photos of it. So you can see this was the inspiration actually of the Alien movie. So it was kind of crawling about, and these are the little babies in this salp. So that was a really cool find. He never saw one, and I knew exactly what it was. So that was really cool. So you can find really unexpected and amazing things if you go look at the beach for a long time. But hopefully in spring it will be more like this, and you can go rock pooling. This Falmouth. You might not be as enthusiastic as I am with rock pooling because I kind of clamber into caves and stuff as well to see stuff. But you can take your kids. So this is my son, and this is on the Scillies, and this is a bootlace worm, which is the longest organism on the planet. So I think longest one was 50 meters. Although I guess they're quite stretchy, so you can probably break the record by cheating a little bit as well. But this was quite long. So there's all kinds of really fascinating organisms, and these are quite common actually.
So you don't need to go to June.
No. This is the Cornish sucker, top left, with these fake eye spots, which is quite common. It has a sucker, so it sucks onto your hand, and you do this, and they hang on. A sea snail, much more rare, also weird little, so it's not a snail, but a fish. And this is a worm pipefish, and just like seahorses, the males carry the eggs, not in a pouch, but in kind of a ridge. And they're also very common species in Cornwall and Devon. But if you really want to find interesting stuff, you have to stick your head underwater. So I advise you to buy a snorkel because it's much cooler. And sometimes it's really nice in the sun, but since you're wet anyway, it doesn't really matter if it rains. So we go out in the rain as well. I often go just to the rock pool, so I don't even have fins on or something because it's not really needed. This is a friend who actually does have fins on. I had one time when I was taking photos of starfish without moving too much, and someone tapped me on the shoulder. He came down from over there because he thought I washed up or something like that. I was live. And that is really great because it might not look like much, and it is a bit cold after 2 hours, but it's totally worth it. So
You probably thought you were trying to get into the country illegally.
yeah, very slowly. So I just have some photos of things that are very small because you have to appreciate the small stuff. So this is a gem anemone and 2 chitons. So these are bivalves, or sorry, these are mollusks, but they're not kind of clams, and they're not snails, but they're a thing that's very different. So they have like 8 overlapping plates and a girdle around them to hold them together, and they graze on the rocks. So these are 2 different species, very small, as you can see. So I cleaned my fingernail before taking this photo, really, but it gives you an impression that that there's very small things. So you need to put a macro lens. Yeah, they're very pretty, but the more kind of you zoom in, the more you see, of course. And you might have found cowrie shells on the beach, little cowrie shells. They look very different when they're alive because their mantle covers most of the shell, and you can see its little squinting eye there, glinting eye, and they're really amazing little mollusks. They're about a centimeter, if that. And what they eat are these colonial tunicates related to the salve. And apart from fish, this kind of crust on rocks or seaweed is actually most closely related to us in the rock pools, because it grows like these colonies where you have these different zooids that have an inhalant opening and then a shared exhalant opening in the middle. But they have larvae with a notochord, so very kind of a tadpole larvae. So they're hemichordates. They're not vertebrates, but they're the thing that's most closely related to us. So they swim away with a little notochord, these starcidians, and that's what these cowries eat. This is the egg-eating nudibranch. So this is a very small mollusk, very beautiful, and it eats the eggs of fish and of squid. And here you see it laying its own eggs. So that was a really interesting find. This is even more niche. So this is also a nudibranch, tiny 1 centimeter kind of long slug, and it only eats the eggs of rock gobies. That's the only thing it eats. So very specialized. Here are all little fish in their eggs, and you can see these serrata, which is kind of an increased surface area to kind of for breathing, are kind of disguised as the eggs with little idols as well. So they're very well camouflaged. That's why people don't see them if they are looking at goby eggs at all. So that was a really nice find. This is not a nudibranch, but it's also a sea slug. I won't get into the technical details of the differences. This eats seaweeds, graze on seaweeds. It's also like a centimeter. But it's a kleptoplast, so it gets its energy from eating seaweeds, but also what it does, it keeps the chloroplasts of the seaweed alive in its own body. So it lives, it continues photosynthesis, so they're truly solar powered animals, which is really interesting, and they're quite common. You have to look for these green sponge finger seaweeds in rock pools, and if you find that seaweed and you look in spring or in summer, you have a good chance of finding these solar-powered sea slugs. This is just a pretty picture of a worm. I don't know what species it is. There's so many species, it's hard to find out sometimes, but it's just to show you that worms can be really pretty. This is a broad-clawed porcelain crab. It's about up to 1 inch across, and they're very flat, and they're underneath rocks, so they scurry away when you turn the rock. But I think they're extremely common. There's usually, if you have a rock, there's 5 or 10 underneath them. But really fascinating. They're so flat, and they're big claws for their size. And this is a pycnogonid, which is a sea spider. has a very, very slender body. It's so slender that not all the organs fit in the body, and the organs are, part of the organs are in the legs, including the reproductive organs. And this is a male carrying eggs. This is a special organ to hold the eggs, just like seahorses, the males take care of the young. So also absolutely fascinating animal. And these are my favorite, maybe stalk jellyfish. So again, these are like a centimeter across. They're not real jellyfish. They're related to jellyfish, but quite distantly. And they kind of catch things with their little tentacles, very tiny things from the water. There's a couple of different species, and they're living only on seaweeds, and they're really pretty. We even have some corals. So this looks quite big, but I think this water is about 20 centimeters deep. And you see two different sponges in the middle, a golf ball sponge at the back and another sponge, and all these little gold things are individual coral polyps. And this is one of my favorites. Few people look at this because it's a little 1 centimeter flabby thing underneath silty rocks. This was actually discovered in Falmouth in Victorian times. And it's just a hydroid, a weird animal. And these are little tentacles. So it can extend from 1 centimeter to 10 centimeters. And it just kind of roves over the rock, trying to find tiny little crustaceans to eat. And all this white stuff is reproductive organs. So really, really weird thing, but I kind of, yeah, gotta appreciate not so photogenic things as well. And then finally, I'm cheating because I took this photo in Wales. You might have seen these springtails, so they have 6 legs, but they're not insects, and they're extremely hydrophobic. They have bristles and have pheromones to attract each other. So they live in rafts in rock pools. So they eat detritus, but they're eaten by mites. So here you see 2 different species of mites crawling over this raft of springtails, and this is about the size of a stamp. This photo. So there's a lot going on. 3 different species. Not uncommon. And then this is the last thing I'm showing, and you can run away. This is something that actually grows up to like 2 or 3 feet in diameter almost. A spiny starfish. So like quintessential rock pool organism, but really interesting if you zoom in. So like sea urchins, echinoderm, derm or skin, kind of spiky. This is very spiky. You can see in between of the spikes, you can see gills. And if you zoom in on the spike, you see these little kind of feet surrounding the spike, which are special organs that pick off sand or other organisms. So they use that to clean themselves with, so they're not colonized and end up being all encrusted. And if you turn them over, they have these hydraulic tube feet, so they're filled with seawater, and they move by basically varying the pressure of the seawater in this locomotion system. These are some of the feet near the mouth. And of course, they need to have this water in that system to move, and they let in seawater. If you look at a starfish next time, see if you can find the madreporite, which is on top. So it's a little sieve. You can see that it works because there's some debris on top. And that is basically to let in the seawater for this hydraulic locomotion system. So yeah, it's really interesting. And then finally, they have eyes as well. Usually British people, I show this. I think it looks like something else. But anyway, so they have.
Use the mouse just to point out.
Okay, yeah.
Yeah.
So every kind of red dot is like a pigment cell. It's like gives 1 pixel of vision. So they don't have very good vision, but it's good enough to kind of separate the sand flat from where the reef starts. So you see light and dark. So these are eyes at the end of every leg, which I thought was beautiful. But anyway, thanks for your attention.
Thanks, Mick. Wow, to paraphrase Shakespeare, "Oh, brave new world that has such creatures in it." That's amazing, amazing. I don't know if anyone else has seen any of those creatures or before, but extraordinary. Thank you very, very much indeed. And Thank you for coming up from Penrith and joining us.
My pleasure.
We could be around for a few minutes if you want to ask some. Sign the register.
Oh, I'm using a Olympus Micro Four Thirds camera. Yeah, yeah. And that in a housing, which housing is very expensive.